लंबोदरस्य विट्टो विघ्नराजो गणाधिपः धूमकेतुर्गणाध्यक्षो भालचन्द्रो गजाननः द्वादशैतानि नामानि यः पठेति शृणुयादपि सर्वकार्यसमारम्भे विघ्नस्तस्य न जायते। ओम शुक्लांबरधरं विष्णुं शशिवर्णं चतुर्भुजं प्रसन्नवदनं ध्यायेत सर्वविघ्नोपशान्तये। लक्ष्मीकान्तं कमलनयनं योगिभिर्ध्यानगम्यं वन्दे विष्णु भवभयहरं सर्वलोकैकनाथम्। अज्ञानतिमिरान्धस्य ज्ञानाञ्जनशलाकया चक्षुरुन्मीलितं येन तस्मै श्रीगुरुवे नमः। ध्यानमूलं गुरोर्मूर्ति पूजामूलं गुरुपदः मंत्रमूलं गुरोर्वाक्यं मोक्षमूलं गुरु कृपा। आप सबको धन्यवाद देने के हमारे शब्दकोश में शब्द नहीं है। आपने जो सम्मान किया है वास्तव में हम उसके अधिकारी नहीं, परंतु फिर भी हम आपको धन्यवाद देते हैं। अब हम आपसे पूछेंगे कि आप लोग हिंदी में चलेगा कि अंग्रेजी में चलेगा कि दोनों में चलेगा? ठीक है। जो आज आप हो वो हम कल थे। ठीक है। तो जब हम यहाँ पे थे तो हम किताबों में पढ़ते थे डीएनए, डिऑक्सी राइबो न्यूक्लिक एसिड, डीएनए आरएनए। तो मन कहता था कि इसको जिसने खोज निकाला है क्या कभी उससे मुलाकात हो सकती? तो इस जिज्ञासा के साथ कोई अमेरिका से आता था तो हम भी उसके पीछे लगते थे, देखते थे कि कैसे चलता है, कैसे बोलता है, क्या करता है। तो जिज्ञासा इतनी थी कि मन कहता था क्या हम कभी अमेरिका जाएंगे। और आज उस परब्रह्म परमात्मा भगवान श्री राम की कृपा से आज हम वहां पे 38 सालों से 38 इयर्स। आप में से काफी बच्चे उस समय नहीं रहे होंगे, आए होंगे उसके बाद। Who am I? ये जानना जरूरी होता है कि आप कौन हो। जीवन का मकसद भी यही है। आपके मन में प्रश्न उठता होगा आखिर हम कौन हैं? Where I come from? Where I go? Who am I? So हम इसको साइंटिफिक दृष्टि से बताना चाहेंगे। इस चित्र में आप देखो, अगर हमको आप पहचान सकते हो। मुश्किल हो जाएगा, हो सकता है कि हाइट निकाल दूं तो कुछ। ये हम हैं यहाँ पे। ये 76 की थी जब हमने मास्टर्स किया था, और उसके बाद हम लोगों तो ये हमारे साथ का अल्ताफ अहमद लाल, all these are our good friends. Altaf, I think still he is in Sun Pharma somewhere, but he is very good scientist. Ashwani Kumar, he became director ITRC and retired. And Raj Kishore Puri, we all used to live in Moti Mahal hostel, having fun and doing research. So Puri is still, he was with me in FDA. In recent past, he left, he retired. I'm still in FDA. And he joined another company. Very nice, as a vice president, executive vice president. Juhur is no more now. Jaiswal is, and Prem Prakash Dubey. Sinha. I think Sinha is still here somewhere. So anyway, to put this picture in front of you to let you know that I am the same person, just like you here, 38 years ago. But there was inquisitiveness, a ambition to know something, to know those people who have discovered something unbelievable. DNA, RNA. So, I'm sure you might be remembering this one. It's James, Nobel laureate James Watson, J.D. Watson. I wanted to meet him when I did go to Chicago. There was a DNA Helix conference in Chicago. There were 43 Nobel laureates at the time. I also submitted a paper, and paper was accepted. I was there to speak. But my curiosity was not to speak too much, but meet these people who have given you a path of whole life. DNA, RNA. Their discovery is still the same. Opened the path of new world, biomedical sciences, medicines, drugs. So I met him. He's James D. Watson. Now, after meeting, I thought if I can have a dinner with him. So I don't mind to talk to people just like you. I approached him. I said, Dr. Watson, I have no words that you have some time to talk to meet, but I need to sit down with you. And he happily accepted my request. So I was with several Nobel laureates to sit down to have lunch. So they gave me a confidence. Now I know closely how a Nobel laureate walks, how he talks, what he does, how he moves, how he speaks. So I got little more confidence. See, in your life also, talking to you today, meeting to you today, I just wanted to let you know you are equally good as me. You are equally good as Nobel laureate Dr. Watson. Then I was thinking to work in the Nobel laureate laboratory. Now see how change is happening. This is me here again. And this man is Mario Capecchi, the Nobel laureate. Mario discovered and gave the world a new technology, gene editing, knockout, transgenic. His associate Kirk Thomas. This is in 1996. And the same time, I was given a responsibility in Northwestern University Medical School to develop a laboratory for transgenic knockout. Just for your information that Northwestern University Medical School is one of the best in the world. Department of Pediatrics is number one. And it has its own unique highness. It also has several Nobel laureates there. So, Mario, see how happy Mario is and how happy we all three are. So I asked Mario, can I have a picture with you? He said, why not? And in 3 weeks, he trained me everything. He said, Rama, now you know everything and you can do everything. One thing, when I reached to his laboratory first day, I told Mario, this man, Dr. Capecchi, I don't know anything. And really, I didn't know anything. Because the technology what he developed, produced, is there any time limit? Because I always like to sum up whenever I am supposed to. So Mario said, no, 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 no. And first day he and I sat down in front of each other, just like a student and teacher. He taught me everything what I wanted, what I'm supposed to. In 3 weeks, I was perfect. People who are here in biotechnology or transgenic area, this is very tough job. And as a scientist, I used to take the challenges. So first thing, you also always ready to take the challenges. So 2 things I want to certainly tell you. You are the master of your destiny and you can do anything. The moment you decide, but don't speak from the lips. Sometimes I tell that people say, I love you. It doesn't matter. Unless it comes from your heart. And then you don't have to speak, I love you. It goes beyond. Right? Likewise, you have your determination. Your determination should be to that extent, I have to do it. So, Mario taught me when I came back, as you learned that I created 27 knockouts, disease model animals for different diseases. And then I invited Mario. I said, I want you to come to bless faculties, students, and me in particular. Mario accepted my request and he came. The whole hall was full like this. And I saw he also did not stand on the podium. He also walked all the way to this corner to that corner. And he knew, he wanted to tell that everybody is listening. And what he spoke, pin drop silence, absolutely, for an hour and a half his lecture did go just like gone like this. So I was very pleased and happy. Mario usually does not go anywhere. I was told that at the last moment, Mario cancels his schedule. So 2 days before I called Mario, I said, Dr. Kepeki, I learned that you cancel your schedule sometimes just 24 hours, 20 or 48 hours. He said, not every time. I said, please don't do this time. And he said, I will come. And all these meetings, all these talks with these people, that gave me, that boost my whole confidence that, wow, now that thing which I used to think when I was doing my master's, that was different. From 76 is 96. See, I also, when I moved to United States Food and Drug Administration, FDA, I joined as a toxicologist, pharmacologist, biologist. This guy is Farid Murad, another Nobel laureate. The same time, I also joined George Washington University as associate professor in Department of Medicine in the division of kidney and hypertension. That's my area. I like to. And that's what in FDA also I'm in the division of cardiology and nephrology. All the drugs approved worldwide that goes to FDA, we are the people who approve the drugs. In particular, nephrology area, kidney, hypertension. So Farid got Nobel Prize in 1999 for his discovery of nitric oxide and its role in severe cardiovascular diseases. So one of my friends, Rakesh, who was professor and head, he was in Texas, and Farid was his good friend. He invited him, and Farid joined George Washington University. And I got more opportunity to talk to spend more time. So when everybody is gone, then I used to sit down with Farid to get more insight. How these people think. So one thing is very important. These Nobel laureates, people, great scientists, they think beyond, not today, beyond. And that's what I like you guys to think also. So that's fine, no worry. I also wrote a paper in, I think, 2011. My area also is epigenetics. I wrote, the title was Beyond Genetics. Epigenetics is beyond genetics. Anyway, so by this time, I developed a confidence that me, Mario, James Watson, Farid, all these are good people. And the difference is I don't think the way they think. So that's what I want to let you know, that think beyond. Not today's problem, but problem which you could address tomorrow. Now I am in FDA. His cardiologist, our good friend, Commissioner Dr. Robert Califf. Califf also visited India, I think, last year. So now you can see that meeting with the people, talking with the people, it gives you a confidence. And you think that, yes, now I can do something smarter, something better. So आज के लेक्चर में हम कोई लेक्चर ज्यादा नहीं देंगे, केवल इतने बताना चाहते थे कि हम कौन हैं। हम आप ही की सीट पे बैठे हुए एक स्टूडेंट हैं, जहां आप बैठे हो, और हम भी कल वहीं बैठे थे, परंतु आज सब लोगों का आशीर्वाद, सबका साथ, सबकी कृपा से आज हम 38 साल से 38 years. It's long time, but I sustained. क्योंकि academia is very tough, very hard. Grants. But since I moved to FDA, now this problem is gone. I am a federal government servant. I work for US government. So that thing is now over. What will happen tomorrow if I don't have grant, where the money comes from? And that was the reason why I moved to FDA. So since 2008, I am in FDA, and concurrently I also have appointment in George Washington University Department of Medicine. I go time to time to grand rounds. Thank you. Consulting, to speak with the students, residents, senior fellows attending. That gives me a confidence and a pleasure that today I am able to talk, to give some direction, something. So that's now, since I'm here from FDA, so how drugs are approved? because in America, you cannot take any medicine that's without prescription. There are two types of medicine. One is available on the counter, we call over-the-counter drugs. And next one is prescription medicines. Prescription medicine you cannot get until it is approved from FDA and until it is prescribed by your doctor. So when we approve the drug, because we are in that process, that was also my kind of inquisitiveness, curiosity. How FDA approves the drug? So we used to give lecture from bench to bedside. I had a class, bench to bedside, how we do the research, and the research outcome is translated to medicine. So I may not go too much in detail, but let's see. FDA must regulate and approve new drugs. The drug approval process is very hard. It takes 10, 15, 25 years. That's why the medicine is very expensive in America. Sometimes 10 to 15 years, 25 years. Sometimes some drugs are discovered by chance and we also approve them, but we want to have a good idea. The main goal of Food and Drug Administration is the drug which goes to public, that must be safe and effective. So it's called safety and effectiveness, efficacy. Two words are very important in drug approval process: safety and efficacy. The drug must be safe and it should be efficient. It should have effect. With these two words, the whole FDA is moving. Rate of approval is very little. In 1 year, 5, 10, because once FDA approves, it gets and the public gets confidence. And American public is very confidence. The drugs are approved in FDA. They have faith. Once we put our seal, they are happy. They can take that. Once they see the FDA approved, they are very happy. So approval process is very tough and very little, 5 to 10 drugs are approved every year. Why? Because it goes from different layers. So first, my involvement is non-clinical. I'm a scientist. Non-clinical means we want to make sure the drug discovered by scientists or pharma company is safe and it works. So mechanism of action, MOA, we want to verify the drug's mechanism of action is correct, what they proposed. In addition, what we do, the data they generate, like animal data. So what happens, the drug is given to animals first. And our job is to look very closely each and every data. How did they generate the data? What did they do? The lab is, we also have a criteria, good laboratory practices, GLP. The drugs, all experiments, all things must be done as per to the FDA approved laboratories. what we call good laboratory practice, GLP. And time to time, scientists like me, our FDA is very big place. 8,000 people working on the same. There are 4 or 5 centers, and everyone is involved. So, first thing, pharma companies were seeking to develop a drug that should go to these scientists. Center for Drug Evaluation and Research, the place where I am, CEDAR. In FDA, there are 5, 6 centers like this. CEDAR is for drugs. CDR is for devices. CVM for drugs that for animals. CIFSAN, that is for food. So all these centers, they are independent centers. So in here, when the drug, any drug comes to the CEDAR, and then it is forwarded to that division where it is responsible for. For example, if it's kidney and cardiology and nephrology, that comes to us, our division of cardiology. So first thing is non-clinical evaluation, because we want to make sure the drug works. So we used to take the most very closely related species. Mouse is very close related. Pathology, anatomy, very close. So most of the experiments we ask a sponsor to do with rat and mice. When the data comes, we evaluate. A sponsor must test new drugs on animals for toxicity, toxicologist. Multiple species are used to get the basic information. We want to make sure that it is not toxic. And then, once we make sure drug is non-toxic, so we have different meetings. Sponsor, they intending to submit application, they submit, we do first meeting, we advise them what they have to do, how they have to do, and what we expecting them from this data. So once non-clinical is done, then it comes to human being, and that is called clinical trials. I'm sure in this hospital, you guys are very well equipped here. You will see some clinical trials sooner or later or in future. So first clinical trials we want to use, we call phase 1 study. In that one, we used to give in healthy volunteers to make sure that very few numbers. That is called phase 1 study, 20 to 80, and we get how we decide the toxicity, we decide pharmacokinetics, how drug is distributed, what organs, what time, how soon it goes to which organ, target organ. For example, the drug is kidney drug, how soon it reaches to kidney and how much, how soon it is metabolized, what are the metabolites, whether metabolites are toxic. So all these things we get and find out in phase 1 study. In phase 2 study, we have another meeting with the sponsors. If the drug passes the non-clinical and drug goes to phase 1, if we find something, we stop immediately or advise pharma company to change the protocol to do some more experiment to convince us FDA people that drug is safe. Because we do not want to put any drug in human body if it is not safe. That is first goal, safety. So in phase 2, we increase number of people 100 to 300. At the end of phase 2, sponsor discuss how large scale studies in phase 3 will be done, because we want to have the data. Discuss just like you and I we call face-to-face meeting. So I was telling yesterday to our Honorable Vice Chancellor what we do in face-to-face meeting. Sometimes we don't speak, but we know what you want to speak. Speaking without speaking. Because when I am with you, your smile, your reaction, the way you do, it gives me an idea what you think. And that's what we have always face-to-face meeting in FDA. So we have this meeting, we have generated our data to tell this drug is not going to go forward, or we ask a sponsor. So we have a lot of discussion. This is our face-to-face review meeting. FDA and drug companies, and we all sit down and talk. But our goal is not to discourage the sponsor. Our goal is to encourage the sponsor and companies, however, making sure the drug is safe. Because we don't want to put drug in the market to give American public or people worldwide. Once FDA approves, it goes worldwide. Lot of countries, they don't need any more approval. They say FDA has approved, we have confidence. Now, in phase 3, goes up to 3,000, 1,000 to 3,000, and we get more data from. And this goes to patient. First phase is healthy volunteers. Second and third is patients, but limited number of patients. Those patients who are volunteering themselves to participate in the clinical trial. And that's what it is. Phase 3 studies, 1,000 to 3,000 people, safety and efficacy. These two words I spoke first is very important. And then, once it goes to market, we also has monitoring system, very efficient. Even one patient dies, we have a right to stop the drug. If the patient death is because of drug. So there is continuous monitoring. 24 hours, 24/7 system is there. And once we have all the things, data done, so first phase 1, 2, 3. The sponsor submits application that we call IND, initial new drug, that never came. And now, once we have all the data, we call new drug application, NDA. Again, it goes to review process. And in this one, we have quality control. We have officers from Office of Pharmaceutical Qualities, Office of Inspection. They go to see the facility, how facilities work. You might have heard that Ranbaxy used to be here, and they did something. And FDA has a lot of rights, fines in billions of the dollars. That company was, I think, sold to Sun Pharma and then somehow still surviving. But the Ranbaxy is now. Our goal is to make sure sponsors' applications is correct, mechanism of action is correct, and drug is safe. And once we all have, it comes to NDA. Now, at this time, we have a time limit. We have time limit at every stage, but this is very critical. In 60 days, we have to determine and make a decision, sponsor. By this time it comes, it takes 10 to 15 years. That's what I want to do. And by this time, the drug comes to this phase. The moment it comes to this phase, the stock price goes up. The moment the sponsor submits NDA, stock price goes fast because now they have chances that drug is going to be approved, almost. Facility inspections, other things. So NDA, as I said, this is something when a sponsor hears that one, they thank FDA, thank you that we came to this door, and that should be the last door. Then we go for labeling. For example, labeling, you see always in the drug, little chit, and we have all the information there. We ask a sponsor to provide all, for example, what are the side effects for some of the drugs? What are the drug-drug interactions? Which drug you should take, which drug you should not take with some drugs. In addition to all this, when drugs are approved, still we have monitoring. In the recent past, we found that, for example, diabetic drug metformin, for example, cholesterol drug, statins, high blood pressure drugs. We found the drugs, they do have some type of carcinogens, and one was nitrosamine. So we came to know to 2018, and FDA got alert. So we asked a sponsor to take out all the drugs from market, and we gave new rules. Now, nitrosamine 26.5 nanogram. We do not approve any drug where nitrosamine is suspected more than 26.5 nanogram. So time to time, what we do, we recall the drugs. Time to time, we look at what's going on in the market. And as I said, FDA also controls food. And for example, last week, it recalled broccoli chips because of the broccoli listeria contamination. So FDA's job is to provide American public good quality food, good quality drug. And if we are monitoring, we recall the drugs, and now the new batch of metformin coming that should not have nitrosamine, that is carcinogen, more than what we approve. So, over-the-counter drugs, we do not go too much in detail, but yes, we ask them to provide us the detailed ingredients. And we have a database, ingredient database. One of our scientists, we check, there should not be more than that, and that's fine. We do not go in too much detail for over-the-counter drugs you can go and buy from pharmaceutical companies. Drug companies must confirm the requirements for the medication. That is what we do for generic drugs. So you might have heard of brand-based drugs. Pfizer is making something, but Pfizer, we give a right that that drug cannot be reproduced until proprietary rules are over. So brand name drugs are more expensive, and that drug, when it has certain time limit, approximately 20 years something, we allow them, other companies, to produce. So it becomes cheaper. However, it has the same ingredients, same effectiveness, but little less kind of, they change something. For example, sodium chloride, saline, glucose, pH. But generic drugs also approved and monitored by FDA. FDA reviewers will approve the application. Once everything is done, then the final thing comes. A letter is being sent to pharma companies, drug approved or not approved. Why not approved? We tell them, this is why we don't approve, because we don't think. FDA's language is very good. We always think. Our thinking is. FDA is thinking. And we don't pose, but our thinking is final thinking, that this is what we are thinking. And that's why we don't believe a letter that you have to change something or something. So all the FDA approved drugs, you can go to the site drugs at FDA, you will find a lot of drugs. So by this time, I do not know how much time is gone, but 50 minutes. That's what we typically want. We don't want to bore or to give so much. So in that sequence, one time I also thought I wanted to go to Stockholm and to give a lecture where all the Nobel laureate ceremony happens, Sweden. So in 2016, Farid recommended my name. He said, Rama, I said, his name is Farid Murad. I said, Dr. Murad, I had never been to Stockholm. I wanted to go and see where the Nobel laureates speak, that podium, that place. And Farid said, I will write somebody. He wrote somebody, he recommended my name. So in 2016, I did go to see that place where I saw all the names of those Nobel laureates. And I said, wow, these are the people, those great people who made a difference in the life of people. So out of you, who knows, tomorrow one of you can get. So Nobel Prize is that where that's the end. Beyond that, nothing you can say wow. So I'm thinking to stop here and give opportunity and your thoughts to listen and answer. If you have any question, you ask any question. I will try to address if I can. Thank you, and thank you.
Benzene, benzedol for the cancer cure. But two of these drugs are, first of all, the benzene benzedol is one of the veterinary drugs used for animals treatment, whereas the methylene blue is one of the most used industrial dyes. But sir, these three drugs are not approved by FDA for the cancer treatment. But in foreign countries or in Western civilization, it is used in more randomness for the treatment of cancer at higher level. Sir, I have read about these three drugs in one of the review papers. Sir, I can not remember that particular paper's name. But secondly, I have listened about it in Joe Rogan's podcast. Sir, what are the thoughts of use of these three illegal drugs not approved by FDA in the cure of cancer?
आपका प्रश्न बहुत सुंदर है, परंतु आपने हमारे इस व्याख्यान में सुना होगा, हमारे यहाँ सेफ्टी और एफिकेसी दो वर्ड जो हैं, अगर दवाएं ये दो वर्ड मीट नहीं करती हैं तो अप्रूवल नहीं होता है। बेंजीन देखो क्या है, बेंजीन इज वेरी टॉक्सिक। I remember one of my colleagues when I used to be here in toxicology center, he used to work with benzene, and after some time he got cancer, liver cancer, and he is no more now. So, for example, another thing, sometimes a drug may be useful for certain things, but our criteria is very different. Our criteria is that making sure, जैसे cancer के patients में, if one patient dies in 100,000, we take the responsibility. So we do not want, he does not want that any person, anyone, by using this, और जब ड्रग्स आप यूज होती हैं तो उनके मेटाबोलाइट्स होते हैं, जैसे हमने कहा कि मेटफॉर्मिन जब लोग लेते थे तो पहले नहीं पता था, नाइट्रोसाम is carcinogen. Now we have recalled all those batches and given them a quantity that any drug, whether it is losartan, whether it is metformin, that cannot be if it has more, and we ask pharma company to reduce the level less than that. So to answer your question, yes, we cannot ask, we cannot give until we make sure the drug is safe.
Sir, if FDA and other bodies have control over the food industries, then why packaged food like chips and other things become the hotspot for the cause of cancer? Why FDA is not controlling over these cancer-causing ingredients like acrylamide and monosodium glutamate (MSG), as we call it in local language, Ajinomoto?
So acrylamide, we used to use, if you run the gel sometimes, PCR gels, other things, we use acrylamide. It's toxic. So what happens, some of the food, for example, food, homeopathic drugs, Ayurvedic drugs, you see FDA disqualified lot of those things. In chips, we also go and see, because I'm not involved in that center. But the bottom line is the CIFSAN, Center for Food, is responsible, and time to time it goes to see what are the edible items they are in the market, like chips you said, biscuits, what, even. So you will notice that these items are in the market, not legally FDA approved, but they are in the market. What we call sometimes, we let it go until somebody complains, and then we notice, then we stop.